Okay, so final talk of the day. So I'm going to talk about this project that we've been doing which we started just uh, after the Delta wave. It's not about vectors, and I'm pretty sure a lot of people might have heard about wastewater-based. Oh, I see some new faces right now. Okay, so but before I start, none of this work would have been possible if I didn't have so many people contributing to this work. Uh, Ansil and Uma have been doing this work even before I started in two STPs in Bangalore. This was just around Delta wave. They have been collecting samples and trying to figure out the protocols and to understand how this works. And just after the Delta wave, then we took over and we started doing this work all across Bangalore. Vishwanath is from Bioenvironmental Trust. He's our water expert for the city. And one thing I would like to say that for wastewater epidemiology, it's just not about the science. You need to have people who understand the water sanitation network, all the hydrology, so that you can make sense of the data. It's just not about detecting a virus or a variant in the city, because this is something we are doing in near real time. We are still doing it to inform that whether you have new variants and what the kind of viral load is for the SARS-CoV-2 and how it can be used for other pathogens. I have a huge team in TICS, which is part of it, including Namrata. She does a lot of bioinformatics for this. And then we have support from Bangalore Sewage Board and also BBMP, who are the implementing partners. So whatever data we collect, or it's usually shared with them, because they are the one who decides whether the mask mandates and everything. So, and it was generously funded through, we have funding from several sources, through some money from ICMR, from Rockefeller Foundation, but also from Tata Trust. Before I go ahead, I didn't introduce myself, who I am and what I do. So I work for Tata Institute for Genetics and Society, and our main mandate is science for society. We do not do any fundamental research as such. However, we do take on projects which make a huge scientific impact, which has high social impact. So SARS-CoV-2 wastewater epidemiology is something which falls into that array. Same for the dengue surveillance, and I lead the disease ecology and environmental surveillance vertical from the organization. We also have antimicrobial resistance and also vector control strategies, anything under that. So we have, and we're also working on rare genetic disorder, on crop improvement, different projects to do with how the crops change, pattern will change with the climate change, but also with the flooding and all kinds of things. And then we have some facilities like insectary, diagnostic platform. So it's a new organization we are developing, finding our way in, while trying to make a difference to science and society. And we are very happy that we are part of the Bangalore Life Science Cluster. So things we do are possible only because we are part of those cluster, because everything works so well, and we have a very nice network of scientists we work with, basically. So we have our own niche while doing all kinds of science we do. Right, so I'm sure you all must have heard of wastewater-based epidemiology. I would be very surprised if nobody has actually heard or learned about it. This is something which has kind of taken over everywhere around the globe since SARS-CoV-2 has been started. For more than, I think 80, 90 countries are doing it, I think even more than that. But it's not something very new. It has been used as an epidemiological tool for polio surveillance, even in our own country. And actually, one of the reasons we got rid of polio from India, because we combined the clinical testing along with the wastewater epidemiology for finding the hotspots and everything, while continuing with the vaccination. And when the SARS-CoV-2 started, we needed a tool in order to understand where the infections are arising, what kind of variants are happening, in different parts. And Bangalore was one of the cities which led this thing. And now going forward, when you know there's a proof of concept that this works and you can find a pathogen, not every pathogens are stable in the wastewater, but those which are, how this as a tool can be used for greater good, for, say, for vaccination purposes, for good research and diagnostic or anything which is needed. Not just for human diseases, but can also be used for wildlife or any zoonotic diseases as well. And I'll talk about that in a little bit in this. So how does it work? It's a very simple thing. As I said, it has been used as an early warning system. So you have people who are both asymptomatic or symptomatic who are infected with this SARS-CoV-2 virus. and you start to do fecal shedding, which gets out, and which sometimes it may not be detected in your, people may not be going to the clinical for testing, which a lot of people are doing right now. There is hardly next to nothing clinical testing going on. So when people are actually, they don't even know they are infected with the virus, you are shedding the virus, and if you are part of a sewage network, your poop will be part of the rest of the sewage network, and it will go to a sewage treatment plant. And that's where someone like us who are picking up those wastewater samples and detecting these virus samples. So around the globe, there are several studies. I mean, there's a whole lot of literature where people have been reporting, oh, it gives you a 5-day early warning, it gives you a 2 weeks early warning, and all kinds of things have been said and done. But it was a very good early warning system, and I tell you why it is not now in a bit. I just want to build up on that, how this helped us as a city to understand what's going on. So Bangalore, as you know, is a very big city. We have around 14 million people, and there's a huge population density. We also have a very huge water footprint also. So around Delta time, when everybody knows there was a very bad time for everybody, was going through, we had no idea how many cases are there, what variants, or what we are up against. So the idea was that can we start looking into some these sewage treatment plants, and the city has 28 active sewage treatment plants in the city, which we started monitoring at the same time. But the idea was not to do an academic study, that it was to set up a system which can be routinely surveilled, but also you can do genomic surveillance, identify the emerging variants, but also make it as a complementary tool, which can be handed over to the government later on, because I don't see myself doing this work forever, and I don't think as scientists, we can define the protocols, we can understand the data and everything, but at the end of the day, as a public health tool, like the polio surveillance was done for the environmental surveillance, SARS-CoV-2 or for any pathogen needs to be done as part of the complementary tool. So that was the understanding or heading understanding we had, and that's how we moved in. So what that meant was that when we are surveying all these treatment plants, we must share the data from the day one. That was the ideology, because you need to build trust, you need to involve multiple stakeholders in that process. You have to make sense of the data and all kinds of things. So it was just not, you got something in your computer and you say, oh, fine, I'm seeing the viral load. Now, in order to set up a wastewater surveillance system, you must know the hydrology of your area. This is very fundamental, because Bangalore being a very, on a 900-meter city, we are on a ridge right now, so the whole watershed system is going like this, which means the flow of all the sewage treatment plants, what it has been designed around the city is according to that. A lot of sewage board people, like the old engineers, they are familiar with the plant, but the new ones have not much idea. But you really need to map all of that. Do you know why we need to map all the sewage network in the city? To define the catchment areas. So if you have all these dots here, they show the sewer lines of the city. This is for the blue lines where the water supplies. It kind of maps very well for the whole city. So somewhere around like 75–80% of the Bangalore is under the sewage network. So if you're collecting data or water samples from here, you have information from what people who are connected to what might be going with the viral load. But what we did, we mapped all the catchment areas, that how many wards are around each of the sewage treatment plant, which also means that when you are seeing a rise in a viral load, say here versus here, these catchment area are prone to higher risk as compared to other, because viral load is very dynamic, it fluctuates pretty much every week, and I'll talk about that in an average of for every week. So these kind of exercise we did while going through, perfecting the protocols, understanding the pipeline with the help of Biome Trust and also the sewage board to understand all those nitty-gritty of things. But we are still not there yet. I'll say why we are not. This is how somehow it looks like. So you have 28 sewage treatment plants. We started collecting them, which are in the very high population density. There's a perennial flow of water. So it's from a scientific point of view, it makes a very good study system also to understand how the viral load is fluctuating. I have 2 staff who goes every day to collect this water. We do it manually. Unlike a lot of developed countries, you have automated sampler where people leave them and you can collect them after 12 hours or 24 hours. We do not have that kind of luxury. So you have 2 people who are doing this work who who go there every day. And there are a lot of things you can do once you have this wastewater sample. So you collect the sample, so it's done every day. So 28 treatment plant. So you have a routine of say Monday to Thursday. Every day the sample comes in from say 5 or 6 treatment plant, but it must be processed on the same day because otherwise the virus would degrade, whatever. So if you want to maximize on that. So the person will collect it, put it on a kind of a freezer box, they bring it back to the lab, we deactivate the sample and we start processing and extraction of RNA and all the things starts. But once you have the RNA from these samples, they are kind of analyzed within 24 hours for using the quantitative RT-PCR. Everybody knows what is quantitative RT-PCRs. It just doesn't tell you that whether you have the virus. It's not just the presence and absence of the virus, but it also tells you to quantify the viral load, right? Which is very useful in a pandemic situation. You can tell the viral load I had last week, has it increased or decreased? So you can model it whichever way you want. Now, one of the also advantage of working on the sewage treatment plant was that you can normalize this data by the population size, but also the flow rate of these sewage treatment plant. This is very fundamental thing because every treatment plant has its own kind of flow rate, which is guided by the population density of that catchment area, which is the number of wards it is catering to, basically. So you can do that, and also you can do something called exponential weighted moving average, and I'll tell you why we resorted to something very simple, because when you are talking and sharing the information to someone sitting in a public health office, you want to communicate information in such a way that it is very understandable for anybody. It's just not that you and me trying to figure out what was the, the load going on and everything. So because it is being used in a lot of stock markets and everything, people know whether it's going high. It's very easy to interpret, basically. You don't know how many people are infected, but from the wastewater, this is an Australian group, they came up with this rough estimated number, this formula where you can calculate how would be the number of estimated people infected in a given time in the city, which is again also very useful. And finally, this was the most important thing which I feel was to be able to define the diversity and abundance of different variants. We live in a city, we live in a country, we do not have a lot of genomic sequencing going on. We have very less data, so this wastewater data is really, really important to understand what variants are flowing. Now, once we have this, so I'll just talk a little bit about how the genomic surveillance, this thing works. So this is based on a pipeline, analytical pipeline. So you have these, the samples which are positive for the RT-qPCR, they are taken forward for the sequencing. It's the same sequencing protocol that is followed for the clinical samples as well. So this is a targeted next generation sequencing for SARS-CoV-2. So there are something like around 100 primers, which are all across the genome of the SARS-CoV-2, amplicon-based. So when you sequence them, when you analyze the data, what it gives you, the single nucleotide variant, basically SNVs, all across the genome. Now, wastewater data is very complex. It's not like a clinical sample, right? One person might be infected with one variant or maybe a recombinant. But wastewater, it's a community sample. You are basically looking at a community data. So you should be able to tell more than one variant in the population. So that makes it even more complex. So this group in UCSD, they came up with this Freya pipeline where what they did was they did this spike experiments by mixing the wastewater sample with the mixed lineage kind of structure, and then they sequenced it. This is just to perfect the pipeline. So what it relies on the abundance, the frequency of different single nucleotide variants, but also the depth, the way it's been sequenced. Once you have that frequency, this relative abundance of each mutation connected to that particular variant, then you can analyze it and say, oh, this sample was kind of infected with this many proportion of this variant. It can fluctuate, and they keep updating the pipeline based on as the variants are going through. So it's very handy. It really has been a lifesaver for lots of places because it's kind of easy to use and interpret as well for the data. So when we started doing that, but also one more thing about the wastewater, I think yesterday, Cliff was showing about how the CT value, this works. So now wastewater also is quite dependent on how many people are infected, right? If you have a lot of people yes?
Yeah, you are mentioning that variants are particular for particular any viruses or what?
Only for SARS-CoV.
Only for
Yeah,
SARS-CoV.
yeah, yeah.
Okay.
The whole pipeline is only designed for SARS-CoV-2. So what they do, they take the whole database of basically of all the variants which have been reported so far, based on because it's connected with the outbreak info and everything. So you have mutations for each of the variants. So if you have those mutations in your database, it will map to that and it will, but it won't just say presence and absence. It will weigh it based on the frequency, but also the read depth, what which you have sequenced it. So it's kind of a I would say, uh, it's a pretty good way of estimating. You're not just arbitrarily saying there was a variant in your.
You are focusing entire this project is on only on COVID.
This is only on COVID.
Okay.
Yeah, this is all
Because
SARS-CoV-2.
previously you showed that polio things
No, no,
also.
no.
That's why.
I never did polio, but I'm saying polio. Oh, should I start the talk again? No, polio was done in India long before, but wastewater epidemiology has never been used for any pathogen until the SARS-CoV-2 came. SARS-CoV-2 is COVID, right? Yeah, SARS-CoV-2 virus. You understand, right? Yeah. Okay. Is everybody with me or any other question? It will get slightly more complex, and I don't want you guys to, I really want you to understand what we have done. This is really interesting. I believe that's what I feel it. Now, the wastewater sample, when you're collecting, it also kind of the viral load fluctuates depending on how many people and what variant actually the city is experiencing, right? So sometimes you have a very high viral load, sometimes you will have a very small window of viral load. When you have a very high viral load, the CT values becomes very small, which means you can sequence very well, and you will get a very good coverage for your genome as well. But also when you have a very low viral load, your CT values are very high. That's very tricky, that point, to sequence those samples. But that is actually also a very important time to understand what variants are there at that point in time, which are actually people are infected with. It's just not the wave when you are going through. This is why I think when the clinical testing is done is usually kind of a rush, like you go and just sequence or test people when who is infected, and then you say, oh yeah, this variant I found. Whether a wastewater-based stuff is more of a consistent way of saying, oh yeah, this is how this is going, and now this variant is transitioning into, and now the other one is coming. So understanding the COVID dynamics, I think this is a much better way, especially if you do not have that much of genomic surveillance based on the clinical data, which in other countries, they have done it very well all throughout the time. So once you have done that, you have the viral load based on the quantitative PCR. This is just for the 6-month data I'm showing you from January 2022 to June 2022. This is the time, so this top window, what we did was we also analyzed this data into 7 days and 4 days 4 days lag. And the reason behind that was that you have so many variants. So the ancestral variant, people said had incubation period of something like 7 days. People will not show any symptom, as opposed to by the time you have some Omicron was coming, it was around 4 days. And you are trying to see how that correlates with this clinical testing, the blue window, which is going all along in the base at the bottom. So this whole thing is for 7 days. This is for the 4 days. When you look at these two graphs, and then this is based on the EWMA that for the estimated individuals. The line here is the mean of the EWMA. So as I said, we wanted to have a kind of a signal system which can tell the people very easily. When the viral load goes above the mean line, which means it triggers a kind of alarm in the system that one should be worried about. If it goes above the 95% confidence interval, which means it's a really high red alert, and I think that is the time if you have a new variant, which could be very infectious, that I think the public health staff, they should be really worried about. But also, when you look at the correlation between the two graphs, our strength of correlation was much better here for the 7 days versus the 4 days. So we carried on with the rest of the analyses based on the 7 days. This is the time when the testing was very good, okay? Now, when you go and you tell anybody that you have a wave coming or you have a high viral load, the first question is usually asked, what is it that driving the viral load? Is it just change of behavior? Change of behavior could be you have, say, a festival or some event might be going on, or like people might have gone back to office, or schools are open, or is it some new variant which has entered in the community? So when we are sequencing these samples, so around this time, this is from one STP, which is over here. It is right in the heart of the city here for Agaram. You can see this is the first Omicron wave that we recorded. This was all dominated by the BA 2.10 variant. Later on, gradually, when the viral load kind of became very less during this period, there was a kind of mix of BA2 lineages. But by the time we had another small surge, which came here, it was all a mixture of BA4, BA5, and all kinds of new lineages which have started to come up in the city again. We did the same thing for other STPs also, and here you have, you can see the viral load is above the mean line, and here it is not going so far higher up. I'll talk about this bit in a bit later, but if you see in generally the composition of the variants, that stays the same. And based on this information, I think it was very useful for the government to understand, should we be worrying about with the increase in cases, because that was the time when the schools were open and everybody was back to work, basically. So there was a kind of concern that why are we seeing so many cases again and what's going on. Now, if you compare this SARS-CoV-2 data, this is the clinical data, this is clinical data versus the wastewater data. The one thing you'll see that clinical, given whatever sequencing we had at the time, this is only for this time period, the clinical sequencing did not record that many number of lineages as such, because it's more of a biased way of doing it, as opposed to the wastewater, because you are getting a community sample, so you basically record more. But in general, the trend stays the same. If you see, the BA2 was recorded here and here as well, except for some of the recombinants were never been seen in the clinical data up to this point. We also analyzed it for looking into the trends of different variants. So this is the original Omicron lineage which came up in the population. So it just shows you that how BA2 has gone down in the 6-month time and other variants kind of starting to go up. We also wanted to understand, do you see any pattern by the location of different STPs? It could be a geographical pattern, but none of that we found. Only the time which was driving the trend in the city. So as a wastewater-based epidemiology, what we set up, it became a very useful tool which tells you that what catchment areas are experiencing high load, how the virus load is going, and what variants are going in the city. And as I said, we had so many meetings and workshops with the municipal corporation just to explain this data, how to interpret, and it's up to them how they want to use it. So they kept renewing a lot. By this time, second vaccination drive was also done. But the only question was that, are you seeing something different from what we may not have seen in the clinical data? And this was combination of the two data sets gives you more confidence, and it was very useful as an exercise. And they kind of accepted that this is a very good thing to do. And even today, if I don't share the data because when there is a lull, I'll get a call and say, do you have anything which we should be worrying about or not? Which I think is a very good thing. Now going forward, lots of people talked about it. We also experienced a very silent wave, which was for the XBB system. There's a talk about how we can scale it up for other public health stuff. Take it for avian influenza, which is such a public health problem from all kinds of reasons. Now, the question I want to put forward here is that we have come this far. It's been 3 years of doing this work, and we have some funding left. We will be doing it. But what does it mean for an early warning system? So what you are looking here is the viral trend that I am projecting is for the wastewater, the data. So you can see this is an XBB wave here, and there is hardly much going on, and there is a JN wave, and after that there was KP1 which came. As I just pointed out, there is hardly any clinical testing going on, and so we can't project any comparison with the clinical data. You can see there's a red flat line, it just stops there. Not even any sequencing going on. Now, under what circumstances the early warning works? Only if you are able to compare it with the clinical data. In the absence of clinical testing, there is no early warning. So how do you define an early warning? How do you say that this was a wave, should not be considered as a wave, differentiating just a signal from a proper well-defined wave? When should you be raising an alarm? I think this is a very important question. And second thing, which is, I think is also from economic point of view is very important, is that we started off with the 28 sewage treatment plant. It's a lot of work. Environmental surveillance, people say it is cheap. It is cheap in grand scheme of things, but if you really want to hand over the program, I think the best thing would be to decide what site would be meaningful, which we can be monitor, say, throughout the year. It could be 10 sites, 20 sites, I don't know how many, but when you see a rise in singular, maybe rest of the city can be surveyed. So these are some of the questions that we are trying to understand based on this data. So to do that, what we thought would be very meaningful is to look at the whole data set now as we go forward based on the catchment area of each STP. So some STP of a very small capacity, which means they are catering to very small number of population size, and some are very large. When you take the data set, say for each wave, so this is a January 2022 to March wave, which I was showing you the trend. You compartmentalize the data and you compare it with the whole city data and you see which size category gives you a better lag, confidence in your lag, the correlation thing. What we see here is that most of the time it was your either large or medium size treatment plants, they come up as the best predictors for a correlation here. So not the small ones. Small ones only come into picture when you have a very low viral load. And I think is the reason for that, there's a delay in the catchment by the time the viral load catches up and it shows up, because number of people, you have to have certain number of people infected to show up in the catchment, in the STP. The other thing which I think is very interesting about all the sewage treatment plants, I just showed you the whole trend. We had 4 distinct waves in the city, right from the BA 2.10, then a BA2, XBB, and JN1. What you see here is that not every STP is showing you a same trend. Some have, like all the waves going on, some are absolutely flat. And what does that mean? It means that not every STP is a good candidate probably for catching the viral load. It could be for any number of reasons. I don't know. I don't know how to understand that, but I think it could be the connectivity of the sewage network. It could be yeah, could be, but when you sequence the information, the viral load is very low. It's not that it's not there, it's just not enough to, this is why I'm saying, what do you call as a signal versus a wave? When I am recording something like this here, I would say, oh yeah, there is a kind of an outbreak or a signal going on. But when you see here, the viral load is very low, but unless you magnify it, but it's not even in par with that basically. So what we're doing right now, we are actually defining taking forward the limit of detection for the virus in different of these STPs, but also visually taking the STPs which are catching most of these waves, that can they be good candidates for as a downsizing the number of the STPs going forward if we were to do this work. Now, if you really compare the whole clinical data in terms of generally how the variants are captured, say in the last 2 years of this work, you see that in generally the trend matches, except as I just said that wastewater does record more number of variants than the clinical data, except that there was not much going on over here. But there are some very interesting features here. So for example, Delta was there, which I think was mentioned earlier that Delta is gone, but actually Delta is still very much there. There are people who are infected with the Delta variant, and you do see that floating around in several parts of the city. But in terms of defining the clinical, the early warning system, this we kind of collected it from the number of people which have been tested and sequenced in the city in the last 2 years. And you can see around the Omicron wave, the numbers were very high and it kind of trailed off as you go now. And if you look at the number of sequences, so clinical sequences, they get submitted to GZ, they are supposed to. It's pretty patchy. In fact, there is a lag of something like going for around 400 days. So if somebody is sequencing, those sequences are not submitted. So if they are not submitted, we can't compare them. They might be submitted 3 months later or something like that. Now, giving that scenario, if you are in, how do you say something which you have find is before the clinical data, or was it appear in the wastewater? So, for example, if you see here, BA 2.10. So if you compare the relative abundance of this in the clinical samples versus the wastewater, you see there is hardly any signature of this variant in the clinical sample, but in wastewater it is there throughout the time, but we can't say where it was because I think it was there already in the population when we started doing this work. But if you look at it for other variant, this is a BA 2.75, sorry, BA 2.86. It was never seen in the clinical samples ever. I don't know. It's a selective sampling. So they may have missed, there will be less number of people, they found it. But it was very much there in the wastewater. And if you compare it with the XBB, this sample is collected, I think in November, something like that. But it was submitted 300 days later. Then we found this variant in the wastewater. And you can see it shows a sharp decline again in the relative abundance as we move forward. So one thing which I thought was very interesting to understand, that comparing with the clinical data versus the wastewater data, can you really understand that how you can project that how the variant is actually cruising? Can you calculate a kind of a growth for that variant? So if you say here, I'm taking only the mutations which are, say, variant has been found, presence and absence, and you do a logistic regression, basically. So day 0, I'm considering wherever it was seen first, because it was seen first in the clinical sample. So what you see here is that from the day it was seen in the clinical sample, we didn't see it until over here, when it was a kind of a good 100 days of lag till it appeared in the population. Do you understand why that might be? Huh? Yeah, so it all depends on the viral shedding rate, how infectious the variant is, but also you need to have a certain proportion of the people infected in a population in order to show up. So by the time a variant infects a certain number of people, then only you will see it in the wastewater. What I am trying to say that the data can only be used as a complementary tool for the clinical samples, but it can't stand alone. If I'm finding this variant, say 100, 200 days later, and by the time a lot of people are already infected, it may not be very meaningful, at least in this context right now. So the clinical testing, there is one sample was submitted. So if the clinical testing has been done on a regular basis, say even few number of people, I think the chances that would have given, look at the shape of this curve versus it's a proper growth it is showing, because once it entered the wastewater, suddenly all the sites are kind of picking it up for the city. So then by the time a lot of people are getting infected, and you see there is a very consistent growth. And this was not just across for the whole city. You can see it across all different sewage treatment plants, basically. And this is another question that we are trying to bridge, that when you are in a city, cities are very important in terms of, that how the variants circulate, because a lot of people, when they come from outside, they land here. The variants are not evolving, or people are coming from somewhere to rural areas. People are coming to Bangalore, then they are going to and dispersing out. So do you see a pattern that does it really spread from the center of the city to outskirts, how that pattern might be? And this is something we are trying to tease apart, that with terms of this, say, XBB, how that was. So for example, this RCL, which is one of the sewage treatment plant, you can see it was much earlier here than compared to some of these STPs where it shows up. And this could be an artifact of that, the size of the STP, population size, or everything. But these are very important things which one needs to understand and take into account. And especially for the diseases where if you want to take action on the ground, especially for things like vaccination or something like different. I am switching to the next part of the talk. If you have questions, then otherwise I'll switch to the thing. Does anybody have any question? Yeah. Yeah, go ahead.
You said that to raise an alarm, there needs to be a comparison between the clinical and this
I'm
one.
saying early warning system.
Yeah,
Yeah,
sorry,
yeah, yeah.
earlier, yeah. But in this case, hypothetically, if there should be an alarm which needs to be raised, the clinical samples are way faster than what is being detected. So how can we raise?
Clinical samples are faster.
Yeah. It's showing faster, and there is a lag for the wastewater.
I
So
know, but clinical samples are only from very few. These are very few data points I'm saying. What I am recording is from all across the Bangalore. I'm telling you even the spread of the variant. Some of these samples may be done, say, in 3 or 4 hospitals around the city. So the spread is very small, basically. And I think by the time this variant, I mean, this is the kind of pattern you see. The buildup kind of takes something like 100 days or something, and I can say for a lot of other data set which we have seen. If it is found in the clinical sample, then it's a, so you know how the clinical testing usually works in the city, that they might have heard there's a new variant has appeared and WHO has made it a variant of concern or variant under monitoring, and then they speed up the whole clinical testing, either in airports or everywhere for the random people. Otherwise, there is no clinical testing goes on. So if they found one sample and then after, say, 2 months later, they are doing some more samples, it's just there. If they knew that it's an alarm, then they should be testing more people, right? Yeah. However, if I had found it a month before, then then they can say that, oh, it actually happened for JN1, which was detected in last December. Uh, there was one sample which was seen by accident. They found on 30th of November, and it was the question asked that, have you seen it? And we have. We had seen it 2 weeks before. It's not because the variant arrived then. It's for us it arrived then because you had enough number of people infected by that time, but because there was no clinical testing. So this is the complementarity I am talking about here. Yeah. And I think it kind of tells you that how the different pathogens might work and they show up in the wastewater. Some might be coming really fast, some may not be. So this interpretation of the data, how useful and it can be used, I think it makes a difference.
You compare it with the test positivity rate.
Where is that data? Where is that data?
You
I
have the
don't
case
have.
data.
I don't, nobody has that data. Do you have? If you have, I'll test it.
I don't have it. But
No,
you're
nobody
working
has.
with the government. You're
No,
supposed to
no,
have the.
no. One of the things which I really want to do is to compare for each sewage treatment plant with the wards, whatever numbers they have tested there, instead of comparing it with across all the city, because I'm diluting my data by doing that. If you do that, then it will tell you where you have better correlation. We don't have that kind of data, basically. I don't know who has and how they can provide it, basically.
Is the sensitivity
Yeah.
is the sensitivity same for clinical testing and the pipeline you have used? Like, is it same in terms of detecting a new strain, or is it lacking there itself?
You mean for the qPCR and all?
Yeah.
Yeah, it is the same. Actually, we started off using the same. So there are no separate PCR kit for the wastewater. We are working on one which has internal controls and everything. So yeah, it works fine. In fact, when we analyze the data, because saying something cutoff of 35 or something is very subjective. So we are we take into account the volume and everything, so we are kind of slightly relaxed in that approach when we say something is positive. But one thing also which I would like to highlight here is that you must have your internal controls when you do a work like that, because what use PCRs, they have a human gene which they use as a control. So we use something, if you get lots of negative samples, we use some kind of a control like PMOV, which is another virus which we kind of excrete as part of our, from our excreta basically, which is there throughout the year. And taking a ratio of that actually helps you to ascertain that there was not a blank sample you had, it was just the SARS-CoV-2 was just very below the detection level. That's why it was not picked up in your sample, basically. Okay, does it make sense? Okay. Right, so we did this SARS-CoV-2 work, but one of the other aspects which we really wanted to bridge was that Bangalore is a big city, but also a city with a very huge water footprint. What that means is that you have all these sewage treatment plants which are treating the water. There is something like 1,440 million MLD of water which gets processed. Yes, you have a question? Yeah. How will you do that? Previous samples.
Actively will test.
From Bangalore? Yeah. How? Who has those samples?
Now we can.
If somebody is testing it, like if the way they were doing the testing, then we can. I mean, they are doing testing. It's not a lot. It's like 100 samples are tested here and there per week, which you can't correlate with this data. I think when the Omicron wave came first time, they had something like 1 lakh tests per day. So if there are only 100 or something or no testing is done, I think the denominator is very small, even to compare. That's why it doesn't matter how much I put that line, it will stay flat, close to 0. But this is not the question I'm saying. All I'm arguing is that if for COVID, viral load tells you that viral signal is going up, but the viral signal is not necessarily going up every time because of that particular variant. Some other variant in the background might be driving it. This is something I want to show here, which, so for example here, this was all the BA2 here, then you had a lean period, then some small sublineages came and they kind of made this here and there wave. You have XBB coming here. If you compare this data with other countries, XBB did not disappear here completely. It stayed here, it became very low. But what we got here is some other BA2 sublineages here. What are those sublineages? We don't know. I mean, they are just called as sublineages because that's what they are matching with the data set. And then it gets your JN1 and other KP1 lineage came here. So this particular point, what I'm trying to say that there must be some regional variants which are very prevalent, which may not be defined as the variant of concern or under monitoring, which are pretty Bangalore specific. And that's something we want to look at, that what that means basically. Right, so in terms of, oh, sorry, yeah.
Yeah, do you all check stormwater drains basically?
Open drains?
Yeah, so this is what I've seen in Bombay, I'm not sure here. We have a lot of stormwater drains in Bombay and they all get mixed with sewage. And they never get cleaned ever. They directly go to the ocean and then fish and then it comes back. So is it something that you guys check as well?
So it's a lot of places where people do not have a very well-defined sewage network. They do collect samples from these stormwater drains or open drains as well. But you still need to know what's the population size are you collecting that sample for. So you should be able to normalize it based on different factors and be able to say that if I am doing this, then this kind of catchment I'm looking at. So there are a lot of studies, for example, done in Bangladesh, a lot of slum populations and all where they do not have, even in Pakistan they have done. What they have done, they have gone and mapped communities and they really defined the system, mapped it using the blue line system and everything, GIS and all kinds of things, and they made these maps, decided what points would be meaningful for what catchment size. So when you have that kind of data for COVID, also for salmonella, typhi, for cholera and all kinds of things, then they can correlate it with the thing. So, but in our situation, if I need to do this work, say for open drains, I can tell you when it will be meaningful. The catchment areas for most of the sewage treatment plants are huge, right? So some of the STPs, the smallest STP have a catchment area of say 2 wards. Still okay, if the viral load is going up, I can say to the public health workers that look, these 2 wards you need to worry about. But some of the areas are like 40 wards, 20 wards. In those circumstances, if you have 20 wards or 40 wards showing this red signal, they don't know where to go. That's where this is why you need a very fine scale sewage mapping for this area. So this work is not there yet for the public health. I'm very honest about it. I mean, it tells you that at say good proof of work to show it works. But in order to make it more meaningful from the public health action point of view, to say imagine if it's a disease where you can control by vaccination, and if you know where the infection rate is high from which area, and if you can say that, look, this is the catchment area you'll be worrying about, like for polio they did it, they knew, and then you can go and vaccinate people for that reason. Or as an ecologist who wants to understand the disease ecologist, not everything is coming because everybody is infected. There are drivers for those diseases, right? Cholera, salmonella. It could be that poor sanitation or something else might be driving those cases. So you want to know what's the cause. Why not anywhere else you have this situation? Why only in this pocket of the city, basically? So those kind of things can be worked out, but you need a very nice, and this is something is our kind of a next phase, as I call it. We are planning to work with the government. It's a huge exercise. It's a Herculean task to do that. But once you have done that, and I think it's just a matter of mapping different pathogens and how things needs to be done, taking it beyond COVID. I don't think anybody is worried about COVID so much at the moment. Yeah.
Got it. And just to be clear, so you're going to map it in the areas where you think there is the density of people, there's a lot of people there, and you're going to do it majorly
So
there.
when I
Just to be
showed the
clear.
first map where the blue line map was, we will follow those lines, and within the wards, you will have to figure out where are those junctions where the connectivity is coming for different wards. So you select the points. It's called something like of an adaptive sampling. You can have, say, within a catchment area, say 50 sites, but you don't have to monitor all the 50 sites on a weekly basis. You can scale it up when you see the viral load or whatever pathogen you are monitoring is going up, and then say, let's do this very fine scale sampling and see which area is contributing more over a period of time, and then you can do it. Yeah. Yeah.
Thanks.
But most of the outskirts of the Bangalore, they are not connected. They are the new developments, like big apartment buildings. So they have their own STPs, by the way. And by law, they have to treat the water before they leave it out, which is something I'm going to mention here, talk about. So Bangalore also has a very huge water footprint where you release a lot of water to these drought-prone areas for agriculture, for groundwater recharging. And it's kind of a circular kind of economy that you have a wastewater going to these, to agricultural fields where people are they're able to grow the crops throughout the year. And as a city dwellers, we are kind of eating all kinds of vegetable all the time, basically. But at the same time, this comes with a cost, which nobody is worrying about, because in terms of wastewater monitoring, when we release the water from the sewage treatment plants, there are very few parameters which are monitored, like in terms of quality, pH, E. coli level, and these are not by any decision, it's by all the pollution board. AMR is definitely never being thought of, not even in terms of any enteric bacteria or any waterborne bacteria which people might be coming or exposed to. Now, just because you can detect a bacteria doesn't mean it's infectious, but it's just a sign that there are risks associated with that. So I just want you guys to keep that in mind. But going by what we heard for the AMR, I think it's a very important and very serious thing, which if you remember Professor Niaz's talk, he talked in great length about how the sewage treatment plants can be contributor for the horizontal gene transfer. It's a very interesting system. It's like, for us, our gut are the main digesting system, right? Where you're digesting food and excreting things out. But for our city, actually, the sewage treatment plants are kind of a digestion point where you are sending all these used water, where the water, with the help of bacteria, archaeoviruses, but they are actually used in these treatment plants as part of the activated sludge to clean the chemicals and pollutants, to make it purify the water. This is a very basic technology. It's used not just here, all around the globe. And what happens is that you have different compartments of these sewage treatment plant. And what this study here, which I have cited here, it shows that at each level of the treatment is a different shift in the community, but also the resistome, which is happening here. And the opportunities for horizontal transfer of the gene is much, much higher. And by the time the water is released from the treatment plant, there is next to nothing way of telling that what it is carrying. Now, if you leave this water in the lakes, or as I think Professor Niaz was saying, that somebody asked that what if you are the sewage, you are standing in a sewage. I don't think it can do any harm, but believe me, whether you ingest it or not, I think we are exposed to lots of resistome in our daily life, especially lettuce and things, which is, if it is grown in this wastewater, there is a transfer of genes through the xylem in the tomatoes, even the lettuce, which is a lot of studies which have shown that. And this is something we wanted to understand at a very fundamental level, that what does it mean for the microbiome for a sewage treatment plant? And before you say anything to anybody that you must change your processing, you have to show it. The only way to do it on a scientific based way, otherwise no data, please don't talk, don't mention anything here. So we did this 16S sequencing for these samples, and what we were really looking for signatures of, say, these human-associated pathogens like Streptococcus, Cyanobacter, Mycobacterium, Escherichia, Shigella, Pseudomonas, Klebsiella. You can see that they are there, except for this, most of them are in a very low prevalence. And if you compare it really from the inlet to the outlet, the abundance is even lower. We try to look at this diversity in the terms of the temporal trend. You see there's inlet has a very different kind of than the outlet of the sewage treatment plant. There are not many surprises here, except for Streptococcus is there, which is not the same species which causes the problem. But outlet definitely has a very rich microbial diversity, and for the right reason, that they are using activated sludge for the cleaning up this water, and that's exactly what you expect. And rest of the thing, I think, are pretty standard what has been seen in other places as well. What we found, I think, was very interesting is that when you look at the temporal trend, say, across the whole different time of year, I was not expecting that there would be clusters. Say, for example, January samples, some of them are clustering separately from rest of the thing. I think it's mainly because, basically, I was trying to understand that does your microbial diversity of your sewage treatment, would it change if you see a wave going through the city? Because people are taking different kind of measures that time, and is it really affecting your microbial diversity? And some studies have shown there could be a effect of that. And June, again, circle corresponds to the June wave at that time. This is all very correlational. I would not say that's because of that, but it does kind of structure in that fashion. We try to look at that there is a change in the log-true change. So firmicutes are basically part of our gut microbiome. They don't really, they are just there, a part of our human poo. So I don't think there is any alarming thing. So we also characterize these different antimicrobial resistance genes. So the whole thinking behind doing this work was that what can you find? Because we do not have any data for in terms of what kind of genes do you see. So as you know, that a lot of antibiotics are being derived from the environmental DNA, and that's how they've been found. But looking at this graph, when I saw it first time, I was actually quite shocked because tetracycline is in such a high abundance here, and so are a lot of aminoglycosides, and they are kind of consistent. If you see this pattern, it's pretty much same throughout all parts of the city. It's not like some are showing less or versus the other. So pretty much it's everywhere. But also we see that richness kind of is higher in the inlet than in the outlet. So there is processing. Whatever processing is being done, it kind of works. It does lead to the reduction. And same for the diversity, which doesn't change so much. And we try to do this linking of antibiotic resistance. So basically what you do, you have this bacterial phylogeny, which has been found associated with different antibiotic class, antimicrobial resistance genes. You try and do this networking to see what shows stronger association with which bacterial phylogeny. This is more of a schematic. It's all driven by the abundance, what you have. It's not something very, so the stronger the line, it shows a very strong kind of correlation. And what really stands out for me is that the inlet shows a very thick and intricate network here compared to outlet. And the only, I think, positive thing I take from this exercise is that I was not expecting it, that the sewage treatment plant, doesn't matter whatever different processing they are using, they do work. They do kind of lead to some kind of reduction in some of these antimicrobial resistance genes, which would also mean that if you change the technology, what they use to a certain degree, say membrane-based or whatever, I think it can go a long way in reducing the environmental impact later going on in the city. We also look for the log2 fold change from inlet to outlet. And as you can see, for some of these aminoglycosides and beta-lactams, there was not much, but for others, it does show, except for the, again, for the tetracycline. So some of the antibiotic classes, they do show a reduction. Some of them are just there all the time, basically. So this is all from the city point of view. I'm not going to show the data from the outskirts of the city. But going forward, I think what we need to come up with is a plan for that. You know that SARS-CoV-2 can be done. Can we select and prioritize some of the pathogens of interest that matters for the city? Do you know how that can be done? This is just a list I'm showing here. No? Okay. I believe if you do not have data on the pathogens and you don't know what to decide, I think the best way is to go back to environmental surveillance. You can do a lot of unbiased sequencing, say different time of year, and you will come up with a good list of pathogens, what are seen and what's going on in your population, basically. We have seen, please don't go by the yellow fever thing, but we have seen pretty much, say, 7 or 8 of them in the different time of year. And one of the things which I think can be very useful is this human papilloma virus. The government of India has recently rolled out the vaccination for that, and I think this is something can be a very good candidate from a vaccination point of view if you target those areas, and it can be done. So something like that will be coming up, along with making the very, I would say, sensitive diagnostic kits, which works very well for the environmental samples. So one more thing I want to highlight here is that just because you can detect a pathogen in the wastewater doesn't mean this is something one should be worrying about doing it. There are lots of things you see in the wastewater, but as I said that you really need to understand the pathogen biology. What does it mean? Can you use this information? I mean, and I think this is something I have been really trying to understand in the last 3 years. COVID was very important. It told you what pathogens are coming. But very recently, there are a lot of studies from Europe and from US. They have been saying that because there is a range expansion in the Aedes albopictus, and there's a dengue outbreaks have happened, even for the malaria in those parts of the world. They are saying wastewater surveillance can be a very useful tool for these pathogens. There's one study which was published which showed that they can detect dengue in the wastewater. How many cases we have for dengue in Bangalore right now? Lots, right? There's an outbreak right now. How much time do you think we find in our wastewater? Never. Never. We don't. And can you imagine if we find malaria? We do. So what I'm trying to say is that this may work for some places as a strategy where you can take probably, so for example, in those European cities where you have very good sewage system, maybe the virus doesn't degrade. For them, it's easy to find, and they can do. This is all based on the symptomatic cases, by the way, not so many active cases, what has been found. So I have serious reservations about that work. But the point I'm saying is that malaria as a parasite is also harbored by other reservoir hosts as well. So if you are seeing lots of that or different kind, I don't think that's a very meaningful thing. For a vector-borne disease, I think for a place like us, I think vector mosquito is the best way to go forward to understand and eliminate a disease or use it as a main system. So not because everybody is doing it, we should do it. One needs to really prioritize what really matters rather than just blindly doing whatever is being done elsewhere. And I think that's the very important thing one should really think about. And finally, what I really believe that sanitation mapping actually is the holy grail if you really want to make it as a very effective system and something that must be done. If anybody, your data is as good as your understanding of your sewage network. Remember that. Just because you're seeing the virus, detecting it doesn't mean anything, unless you can take it forward and use it for some kind of action and planning for that, for the healthcare system. We want to expand it to the different peri-urban areas, and having a sampling framework in different niches. So for example, around the poultry farms or other places where you can look for different viruses if it, just to understand how that works. And the real thing which I believe, as you have asked, that how sensitive it is. When COVID happened, we didn't have many kits. We used whatever was available. We worked with the protocols which were cost effective, say PAC protocol, but also less time consuming. What we really need is very sensitive diagnostic kits. Kits which can detect a panel of different pathogens. So in weekly basis, is the influenza A is going up or B going up, or is it RSV which is up against or different? So like a respiratory panel, a panel for say acute febrile illnesses. I think that would be a very game changer. And if you remember, Dr. Arun was talking about this infection in the forest area where the animals are coming. That is something where this kind of diagnostic would be very useful, where you have so many animal species are coming, you can't test and understand, but just taking few samples on a regular basis, especially in combining with the camera trapping, if you sequence those samples, you will have very good data to understand what kind of wildlife infections are actually there in that population. So you will not have to worry about saying that that animal is found. You can do something about it, basically. So surveillance actually is very, very important. If you don't know, you don't have to wait for those infections. This is why I was surprised that why Nipah virus has not been found in other places, but because places you have good surveillance, you see pathogens. Otherwise, it's as good as it was not there. I'll take questions if you guys have. Thank you.
Information that RNA is quite stable even after a few days.
For COVID? For
Is that
SARS-CoV?
naked RNA or within the virus?
Are you talking about SARS-CoV-2?
Yeah.
So
It
it's
was there
stable
in our slide.
Sorry?
In our slide, it was there.
Is it for the dengue you are saying?
Not.
This one?
Yeah. Up to 21 days.
No, so what
Cold
it is...
storage.
Huh? No. So what it is saying is that, when people are shedding the virus through, mostly dengue is through the urine, not so much through the fecal route. When it is there in the sewage, it can be stable for 21 days. It won't degrade very fast. That's what this study was saying, basically. COVID also, for a month, it will be there. Like, it doesn't degrade that fast. So these are like the fragments of RNA, right? So.
But RNAs are very sensitive for...
It's true, yeah. But as I said, right?
In what sense it is?
This is why I'm saying, if you have few people kind of excreting, that you won't find it, but you have tons of people, like for COVID, you will find it because there is somebody contributing to the problem. Yeah. And that's for a lot of other viruses also, adenovirus, norovirus, and everything. And hepatitis actually is a very good candidate to see that as well.
While doing research, people are thinking isolation of RNA itself is a challenging task. But here, even in the open place, in water, that too, liquid, still it is very stable.
This is why I'm saying that for dengue, we had very little success, not much. We have seen some Zika, but not chikungunya also. Yeah.
So maybe Farah, you talked briefly about how you've been working with many groups through this process. So maybe you can just talk very briefly about what were your experiences, what was the challenging things to communicate? Was it just about being there for long enough? Like you said, now they call you and ask you, if they don't see something from you. So what were some things which were, it took some time to break, some, some experience.
No, it was an experience I can never forget when I did this work in the last 3 years. So when we started this work, that was around August 2021, just after the Delta was gone. And it was a time that everybody, like the whole government system was under a lot of pressure, firefighting kind of mode, right? So when you are building up a program like that and you go with any amount of data, neither the commissioner, nobody has time to understand why are you here and why are you showing this thing. But they will not say no, that this, what it means. It took us 3 months to set up this program, from collecting the sample to figuring out the viral load to running the pipeline for the genomic surveillance, 3.5 months. Once you have all the dots, then you can say you have a very comprehensive data from your samples and you know what variants are there. There were other groups. There was one more group in the city who was looking into 46 open drain sites. So we are doing sewage treatment plants. So you are covering something like 80% of the population, as I'm saying. Open drains are very scattered sites in the city. So one of the challenge, which I would say, or something which one needs to be mindful that everybody wants this to be part of a public health care system. So the government can use it, right? And that was around the same time when WHO also came up with a booklet saying environmental surveillance can be a game changer for a lot of pathogens. When you go and talk to the government people, going as a group really makes a difference, because having 3 groups talking the same thing or 2 groups doesn't really help them. That's the one thing. But for us, the real game changer was that when we called for a workshop with them, where there was a lull period when they are in a mode that they can find, okay, we have some life, tell us what you want to tell us, showing them the genomic data. That was the game changer, that if you are saying the viral load is going up, what did you mean? What are those variants? How many copies? What does the thing? How many people could be infected with the virus? You know, all that information. We never say that how many people are infected to scare people off. It can be, but with the government people, we always share this kind of thing. So those are the learning things which we learn. But most importantly, how do you sell or you communicate science in a very simple way, which anybody sitting in that office can understand? That I think is very, very important. Yeah. Okay. Thanks. Oh, yeah, you have.
Can we use this data to predict the risk zones and suppose to contain if any future pandemic is there, similar type, then we can prioritize those area for vaccination as well as for containment?
Yeah, I mean, if we were doing it before, say, Delta wave that time, and we had this kind of information with the good sewage map, as I'm keep talking about it, this would have been immensely useful. They would have known that these are the areas which are experiencing very high. Let's have a lockdown there first versus here. As I said, by the time we were running this program, we were already gone through the first or second vaccination wave. So that risk factor was gone. The only worry was that what if you get something like Delta again? Because every time when a variant comes, everybody's worried that is it as infectious as Delta or a bit mild? And that is all based on the information or research done. But the fact you are seeing in the wastewater, what should I be doing? Like that's what the municipal corporation will ask, right? Should I be worrying, or should I be increasing the number of beds in the hospital? Should I be again going for a mask mandate, or should I shut down the school? You know, those are the real questions they need that information for. This is why I think this information is very, very useful, yeah, for any pathogen if you spot it, basically, yeah.
And there is variation in data because this disease, when it was in progress or now situation is like normalized, so we maybe the predictions can be different, like because lockdown and other things.
With respect to COVID, you mean?
Yes.
Yeah, I mean, we rely on the that EWMA as a model, and we do it for each catchment area separately. As I was saying, this is why I think we also have time to think about those things in very great detail, that what are those good sites, why the other places are not showing in a better way. Yeah, definitely. Yeah. We are still doing it, yeah. Right now, the viral load is slightly lower, but you see lots of variants even now, basically, yeah.
Okay, thank you so much, Farah.